Good morning, everyone. My name is Andrew Crockett. I'm the CEO and co-founder of KalVista Pharmaceuticals. This presentation also contains forward-looking statements. Just a brief overview of our company. At KalVista, we've been focused on the discovery and development of small molecule protease inhibitors since our inception, and we have expertise particularly in the role of plasma kallikrein in disease. We're developing a number of small molecule plasma kallikrein inhibitors for the treatment of hereditary angioedema, but we also are working in DME and recently announced a deal with Merck for our intravitreal and oral programs in DME, which I'll come back to soon. We're a public company and listed on NASDAQ as of last year. A quick overview of our portfolio. As I mentioned, a number of compounds we're working on for the rare disease of hereditary angioedema. We'll spend most of our time today focused on our work in diabetic macular edema, where we have a small molecule plasma kallikrein inhibitor, KBD001, as well as follow-on oral therapies, oral plasma kallikrein inhibitors. DME obviously is an important disease with the epidemic of diabetes. We're most interested in those patients who are not responding well to current therapies, in particular the VEGF therapies. We have identified plasma kallikrein as an important mediator of VEGF-independent disease, and that's where we focused our work since our company got started. This is some interesting data coming out of the DRCR Protocol I study, one of the largest studies we have with Lucentis over 3 years. And what we think is particularly interesting about this set of data is that you certainly have a very robust response to the VEGF inhibitor in a proportion of patients, but there's also a large percentage of patients, approximately 40%, who are not receiving a very robust response at all to Lucentis in this trial, and that's where we're focused at KalVista. So this data would suggest that there are at least 2 mechanisms in play here, one of which is driven by the VEGF, and we presume that those patients who are responding well to the VEGF inhibitors, this is their primary mechanism. But there's also at least one other mechanism, and we would suggest that this mechanism is leading to retinal vascular permeability, which is not being effectively managed by VEGF blockade. Our group identified the serine protease plasma kallikrein and its release of hormone bradykinin as a central mediator of this VEGF-independent retinal vascular permeability. We've also shown through work over the past 10 years that we have increased levels of plasma kallikrein in the vitreous of patients with DME, and that this increase in plasma kallikrein leads to retinal edema. Here's some interesting work. As I mentioned, we have several independent points of data now showing elevated levels of plasma kallikrein in the vitreous of patients with DME. And on the right-hand side, we've examined more closely one of these cohorts of patients, the Japanese cohort, where you start to see 2 groups of patients that start to emerge. The black bars on the graph on the right-hand side represent levels of VEGF in the vitreous, and the gray bars represent levels of plasma kallikrein. And what you can see in one group is you have both elevated levels of plasma kallikrein and VEGF, and then another group that has primarily elevated levels of plasma kallikrein. These biochemical data might start to give us some insight into some of the heterogeneity we're seeing in responses to the VEGF therapies in the clinic. The role of plasma kallikrein in retinal edema has been very well studied and published. I'll just show you just 2 pieces of data here. On the left-hand side, you have injection of plasma kallikrein into the vitreous of rats, which leads to significant thickening of the retina, which is shown in the red here in the OCT scans. And on the right-hand side, some really compelling data showing that plasma kallikrein gene knockout has a protective effect in diabetes-induced retinal vessel leakage. And we also showed some similar data using a small molecule plasma kallikrein inhibitor. Again, similar protective effects to the knockout. So we're moving forward with KBD001. Again, this is our small molecule intravitreal plasma kallikrein inhibitor. It's potent, it's selective, and I think one of the most important things for us is we were able to show with a small molecule some pretty impressive intravitreal pharmacokinetics, both in rabbits and mice, leading out to post 10 weeks of pretty impressive exposure. We've moved KBD001 into a phase 1 trial. We did these trial in patients who had a suboptimal response to VEGF therapy. The data showed that the compound was well tolerated, but also had some pretty interesting pharmacodynamic data coming out of the study as well. These data here on the right-hand side just show mean data across the 3 doses, but some interesting data, albeit without a control from a phase 1 showing again that plasma kallikrein may be having an effect in these patients who were not responding to the VEGF. We recently announced a collaboration with Merck where we granted to Merck 2 separate options covering both our intravitreal program as well as our follow-on oral programs with some pretty significant economics. KalVista maintains control of the programs through or beyond proof of concept, and then Merck, once it exercises its options, will take over. We're moving KBD-001 into a phase 2 trial with multiple doses now. We'll be looking at repeated injection, 4 injections over 3 months, and we'll be really interested to see the visual acuity we see in these patients, again, who are not responding to the anti-VEGFs. We think at KalVista, one of the things we've been focused on since our inception is the opportunity for oral therapy in DME, and we think it really has the opportunity to be game-changing and change certainly the pharmacoeconomics in this space. And we're really excited about the opportunity here. We just have some data showing actually that an oral plasma kallikrein inhibitor has an effect systemically delivered on VEGF-induced retinal vascular permeability. Again, starting to tease apart some of the interesting parts of these 2 pathways. Thank you for your time.